Hello and welcome to today's PIR live event webinar brought to you by Partners in Research Canada. My name is Stacey Joyce and I will be your host. Today we have our guest, Dr. Thomas Merritt, with us. Remember that you can ask him questions throughout the presentation, and I will make sure to relay those questions to him when we get to the question and answer portion. Let us know in that question text field where you are from, maybe what grade your students are, etc., and we'll give you a shout out. So without further ado, I would like to introduce you to Dr. Thomas Merritt. He is a professor and Canada Research Chair in Genomics and Bioinformatics at Laurentian University. I'll let you take it away there, Dr. Merritt.
Great, thank you for the introduction. Thank you all for joining us this afternoon. It's wonderful to have you here. So I am a geneticist. I'm a professor at Laurentian University, and I am coming to you this morning, or this afternoon, sorry, from 2 kilometers underground at the SNOLAB Neutrino Observatory and Astrophysics, Particle Physics Lab. We've talked, I think in the past, we've had a live event about what I do as a geneticist, and we do some really interesting work using fruit flies, like the flies that we have here. We study genetics, metabolism, the connection between genetics and metabolism, and a lot of really fundamental questions in biological diversity, biological complexity, genomic complexity. And fundamentally, we go from how you transfer or translate from diversity at the level of genes to biological complexity. And one of the things that we do a lot of is we look to see how genetics modifies how organisms respond to their environment. So we do a lot of work with oxidative stress, we do some work with temperature. We've done some work with things like different diet and desiccation, dehydration, that kind of thing. One of the things that we realized a few years ago was that we are near this lab, SNOLAB, and SNOLAB is a fundamental, or is a it's an amazing place for fundamental particle research. It's the home of the 2015 Nobel Prize with work on the neutrinos, but itself is a really interesting environment. And one of the big issues in Sudbury, where I'm living now, and in Canada in general, is deep underground mining. And deep underground mining has a number of challenges to mining companies, to miners, and not the least of which is the physiology of being deep underground. So a couple of years ago, we started thinking about how we could take the work that we do in flies, where we look at really interesting fundamental questions in biology and how those flies and how organisms respond to their environment, and see if we could apply those into a mining setting with the ultimate goal of trying to understand the physiology, the biochemistry, the metabolism of mining. And to do that, we needed a mine environment. And that mine environment for us is the SNOLAB. And so I can pick up my camera and see now, that's a hallway behind us that runs about 150 feet. That's John Hick. Wave, John. That's John waving, who's an undergrad working on the project, and we go along here. It's hard to picture just how big this space is, but you could easily drive a tractor trailer down the hallway that we're in. And we're in a clean lab, which is why I've got on a head net and a bunny suit, and we're in a mine, which is why I have on a hard hat. So it is a sort of, it's an odd place. It's an odd place in many ways. As a geneticist, you don't spend a lot of time in mines. And as a miner, you don't spend a lot of time in a clean lab. And we are spending time in a mine clean lab, 2 kilometers underground, using fruit flies to understand the physiology of mining. And there's some really interesting things that are coming out of this, and questions that we're being able to ask, and some really interesting questions that this is leading to. But fundamentally, what we want to understand is, how does the body respond to being deep underground? So when you go underground, things change, the temperature changes. And temperature is a funny thing. It's a difficult thing for us to model. And so at the moment, we're keeping temperature the same, and it ends up that the way that SNOLAB is designed, the temperature here is just like the temperature on the surface. And so our control, our flies that we leave on the surface, sorry, it's also an active mind, so we have to be careful if they do things like give us announcements on having to evacuate. We have to pay attention to those, which is also something that we rarely have to do in the surface lab. And so when they come over the loudspeaker, we have to pay attention. So we're in this environment that mimics the surface, but it's 2 kilometers underground. And when it's 2 kilometers underground, the pressure changes. And so we're seeing changes in the physiology of these flies. So we do something called metabolomics, where we look at individual metabolites. Except we don't look at it metabolite by metabolite, we look at thousands at a time. And so we've got a instrument on the surface called a liquid chromatography mass spectrometer, and it takes a complex mixture, which in our case is a fly, and runs it through that and separates it out into individual metabolites. And we can show using that about 10% of metabolism changes when you come underground. And what that means from a mining perspective is we know that mining is stressful. In fact, we know that most workplaces are stressful in one way or the other. And what we want to do as sort of responsible employers is make that mining environment as least stressful as possible. And to do that, we have to understand where the stress is coming from. So what is the change? And so what we're doing with flies, because flies and humans share fundamental metabolism, is we're looking to see how the biochemistry of a fly changes in response to going underground, so that we can make suggestions to miners, to mining companies, to say that when you're underground, this is how your body's responding, and how can we work with that to try to make a healthier workplace. So if, for example, protein metabolism is turned up or turned down, is there a way that we can modify that so that there's less stress on the body when you're working deep underground? And so we started this project a couple of years ago, and we've got bits and pieces of that we're putting together now, and very much an ongoing work. But this is just one of the many projects that's going down here. So most of the work in SNOLAB is working on particle physics. And we do that because of the fact that we're deep underground, we're shielded from radiation. And so what they can look at here are small particles that are not visible on the surface because they're swamped out by the amount of radiation that's up there. And so the reason that SNOLAB exists is because it's this very shielded environment. To be shielded, you have to be very deep. When you go very deep, you're under very high pressure, and that leads to the interesting questions that we can ask about what's going on with deep pressure or deep underground biology. And one of the challenges in current mining is how do we go deeper and deeper? And as we try to find resources and better harvest those resources, how do we make a healthier workplace? So I'm here to chat a little bit about the genetics, but Brenda is going to join me. She's been a SNOLAB employee for the last 10 years.
10 years, yeah.
And what I asked Brenda to do there
Hi, Brenda.
is to talk a little bit about what it's like to be in SNOLAB. So this is an unusual place for me. I'm used to working in my lab on the surface, where we've got things like outside and windows,
Yeah.
and we're now...
We need one of those periscopes.
We need a periscope. So we're now 2 kilometers underground. So you've been here for 10 years, and you've seen
yeah.
SNOLAB span from the original construct to where we are now.
Yeah, I was a part of the expansion, actually, painting and...
So what is your role here in SNOLAB?
Cleaner maintainer. I mainly work with the car wash.
Okay.
Nothing comes into the lab without going through the car wash.
Yeah, all right. So
Yeah.
this is one of the things that I had to learn when I came down here and started working as a geneticist. These are my flies. I've got my computer, we've got our little treadmills and things. But when you bring things in, what's a car wash? And then why do we need a car wash?
You clean items before it comes in.
But why do you call it car wash?
That is a strange name to choose.
Okay.
Because there's a car washer and you could just
But
spray and...
It does actually sort of look like a car wash,
Yeah.
but it's at the bottom of a mine.
Well, maybe because the rail trucks are
Yeah.
the cars
That are
are
coming
coming in through the truck. Yeah.
So I mentioned the fact that the reason that SNOLAB is here 2 kilometers underground is because that 2 kilometers shields us from the radiation. There's so little radiation here that dust actually brings in a substantial amount of radiation.
Yeah.
And so one of the things that the reason we're wearing bunny suits and hairnets is to limit the dust.
Yeah.
So your job is to actually literally clean everything that comes in here.
Yes.
So I mean, if you could look down the hallway behind me, there's a lift truck. There are forklifts down here. Every single one of those has been cleaned to the point
Yes.
that they're dust free.
Yes.
And that's just amazing that
Yeah.
you can, one, clean something to that level at the bottom of a mine and keep it to that level. If you walk along the edge and run your finger along these top surfaces, they come back clean.
It's cleaner than an operating room.
And to get here, you take an elevator, 2 kilometers straight down the cage.
Yeah.
It takes about 10 minutes to go 2 kilometers, essentially 1 mile.
Less than that. I think it's about 3 point some seconds because you're going like 1,500 feet a minute.
Yeah.
Yeah. Going quite quick.
So everything that's in here came down that elevator.
Yeah.
Everything that came down that elevator went through
Trammed through the mine.
almost a mile, a little bit over a kilometer,
Yeah.
through an active mine shaft, through the car wash,
Yeah.
and you've cleaned it up.
Yeah.
And then by the
Yeah.
time it comes in here, it's cleaner than an operating room.
If it's in our blue box in boxes, it'll be double bagged, where we'll rip a bag off right to the other side, like the other car wash. There's 2 car washes, and if it's a big steel beam or something, we'll just power wash it and scrub it down, and then rinse it and then dry it and crane it, get it on a flatbed cart. And depending on the size and what it is, on how we process it.
Yeah. And that goes on with everything that comes down here. So John and I are down here as a couple of biologists. There are a number of other labs that are here. I'm not sure how many active experiments we have going on here.
Mini-Queen, Gamic, Pico.
There's somewhere around 10 active experiments right now.
Yeah.
But we talked a little bit about this before. How many people are actually working down here in SNOLAB?
And it varies every day because people are Tuesday to Friday or Monday to Thursday. So Mondays and Fridays are quieter, but you could have as many as 70 and probably as little as 5.
Yeah.
And it depends on afternoon shifts are quieter.
And we just had an all stations call to the refuge where everybody here goes into the refuge for a short period of time and then comes back out.
And we're doing some maintenance on the cage. If there's no cage, there's no work.
Right.
Because if you get hurt, you can't get out.
You can't get out.
That's right.
And so we had maybe 40 people in the refuge just now.
Yeah.
And it's pretty impressive to find that many people suddenly in the lunchroom, essentially.
Yeah.
And to know that many people are working down here day in and day out.
Yeah, it's so widespread out, you don't see everybody, so you don't realize. Yeah.
So from your perspective, you've been here 10 years. What's been the biggest change over those 10 years?
Well, just the size of the place, really, because everything else remains the same. Like, you still come down the cage, still walk in, still take a shower.
Yeah.
You still clean everything the same,
right.
As it did back in the day.
Yeah.
Just, it used to be 20,000, square
right.
feet. Now it's like 8.
Right. Do you know the biggest chamber here?
Yeah, probably the cavity where the AV, the Snow Plus AV cavity is.
Yeah. And do you know what the volume of that is? I know I'm putting you on the spot.
I don't.
You could just about hide a basketball court in this cavity. And it is
I think the volume of the whole lab is 1.6 cubic meters.
No, not 1.6 cubic meters.
Million.
Yeah,
1.6 million
cubic meters. 1.6 million cubic.
Yeah.
The size of the space is truly astronomical. And you come down in an elevator, a cage that's the size of a big industrial elevator. And then you walk out into a mine shaft that looks exactly like you think a mine shaft is going to look like. It's mud on the floor. There are train tracks through the middle of it.
Yeah.
All the walls are wired together. You walk through that for about half an hour. And then to get in, you have to shower. You have to put on a head net. You put on a bunny suit. You get all that dust down because it is absolutely crucial. No radiation, no dust in the lab. Okay, fine. I can handle it. I mean, conceptually, I know what that means. But then you walk into SNOLAB, and it just keeps getting bigger
and brighter and cleaner.
I mean, like I said earlier, you can drive a tractor trailer through most of these hallways, and suddenly you're in a chamber that is, it's like 200 feet high.
Very impressive.
I took a picture of John when we were down here earlier this fall, and I went to the bottom of the chamber, and he came into the side access, and he's this little tiny spot looking off in the distance.
Yeah.
And that cavity is ultra clean and 2 kilometers underground. It's just absolutely mind-boggling.
Well, it took us about 3 years to paint this whole expansion, and then we had to go back and clean it, hand wipe everything you see.
Yeah.
So it took about 3 years from beginning to end.
So the walls behind us are a mine shaft, and mine shafts are just cut into the granite, and then they're covered up with metal mesh that's bolted in to stabilize that.
Their shotcrete was troweled, though. They brought in the from Montreal.
Yeah. So they take a mine shaft and they smooth it off, and then they cover it with this low dust epoxy paint, and then you have to clean all of that.
Yeah. It was fun painting it, though, like all the tips and curves, and it took us 4 months to paint the cube hall.
One room.
Yeah. That was the Q hall, yeah.
4 months.
Wow.
That's a big room.
Yeah. Then we're on that dental machine to do it. Yeah.
And then, so the lab is active year-round. So you're
Yeah.
here. There's somebody down here almost 365 days. Yeah. So I know that we're not going to be here Friday
Okay.
because we're the 23rd. We've got a
Christmas.
holiday, and then
That's right.
we're off for the weekend. And I think people are back on Tuesday.
Yes.
It's just the amount of work going on here is amazing.
Yeah, a lot of the experiments are remotely controlled too, so
Yeah.
you don't really have to be here to run your experiments. And,
That's right.
Yeah.
We're going to figure out a way to do that with the flies.
Oh yes. Okay. No kidding, eh?
All right, that's awesome. Was there anything else?
Yeah. Well, to experience a rock burst down here is quite different too. You know.
All right, so explain what a rock burst is.
Well, we thought we had a rock burst one time. Went to the went to the refuse station, and then I come back to surface, and I'm watching the news, and they talked about an earthquake. Like, you know, I thought it was a rock burst.
So rock bursts are these great big explosions,
Yeah.
and it's the rock settling around you.
Yeah.
And you can imagine that if you're up in the surface and suddenly there's a big bang and the world sort of settles around you, that can be a sort of
Like, whoa, what was that?
an eye-opening experience.
Sometimes it sounds like like one of those skyjacks falling right over.
Yeah.
You know, like it's really loud.
And you're in a mine, and the walls around you are moving enough to sound like it, well, not to sound like, to actually be explosions in the rock.
One time I felt like I moved a little bit.
It's an amazing place.
It can be
to work.
intimidating for some people. It should be for most, but
Yeah,
yeah.
that's awesome.
Yes.
All right, how are we doing? Do we have questions from our class audience?
Yeah, we've got a few questions here. We were talking a lot about radiation. And so I think the typical way we think of radiation is as something that would harm our own health. But maybe you can explain how the issues with radiation at SNOLAB might be different than that.
So you want me to give a shot at
Go
that?
for it.
Okay, so there are a number of really interesting answers to that. So all the world around you is constantly being struck by radiation. And so basically radiation is coming out of the sun, radiation is coming out of the different stars, and it's streaming through us. And it is there 100% of the time. And that is our normal, okay? But what it means is that really low energy particles are really hard to see because they're sort of washed out by all the high energy particles. And so what happens as we go deeper is those high energy particles are getting filtered out by the rock, and only the really small particles are making it down into SNOLAB. And so what the astrophysicists that are working down here are doing is designing detectors that when one of these itty bitty little particles, and these are fundamentally small particles, strikes an atom in these detectors, you get a incredibly small burst of light. And they can measure this burst of light, and they can use that to go back to understand these really low energy, extremely small particles that are the only radiation that can make it down into SNOLAB. And fundamentally, that's how a lot of the astrophysics experiments are set up, is they take advantage of 2 kilometers of Earth to filter out these larger, shielding. And yeah, we think of it as shielding. But one of the really interesting other biology projects down here, so SNOLAB has been here, are we in our 3rd decade?
Yeah, it's been 20-some years.
So there's traditionally the focus has been on small particle research, and recently my lab and another lab at the med school have been part of a push to expand that. And so there's a project called REPAIR, and they're looking at DNA damage and really low radiation. And we often think of it, as you were saying, the idea that radiation strikes things and damages things. Well, there is a natural system in our bodies and in every organism's body to repair the damage from radiation. And so the background radiation on the surface is repaired by these mutation correction systems. What they realized was that in really low radiation environments, those normal processes are actually turned down. And they can result in damage to the system because the underlying basic mutations that are happening are not being repaired. And so this lab is using the fact that SNOLAB is an ultra-low radiation environment to see what happens to organisms when there's even less radiation than we've evolved to experience. And what they're finding is that there is a certain amount of radiation that's actually healthy because it turns on these radiation repair systems. And in the absence of that baseline of radiation, you get more mutations than you expected. And so you might think that the less radiation, the fewer mutations, because you're not bombarding that DNA with these high energy particles. But at a certain level, you need a certain amount of that radiation to turn on the repair. And if that's not present, you actually get more changes, more mutations than you would on the surface. And it's a really clever experiment, and it's running about 20 feet from us here through the double doors. And eventually the fly experiments are probably going to rotate around to share a space with that. But it's a really neat idea. It's a very clever experiment. And it's an interesting way that, like the fly experiments, we're moving out from fundamental particle physics to fundamental biology, fundamental genomics, and taking advantage of the huge investment that we have here in SNOLAB.
Exactly.
Yeah. So what else?
Excellent. What is something about doing experiments at SNOLAB, other than the size when you got down there to see how big these tunnels actually are? What surprised you, being a researcher used to doing things on the surface?
Well, there are a lot of surprises for me. What was your biggest surprise when you started working here?
Just that it's a big, clean environment in the middle of
Yeah.
the major.
Yeah, I mean, you walk through this mine shaft, and it is a really dirty mine shaft.
You don't expect to see what you see when you get here.
And you walk through the doors, and it's really surprising. I mean, there were a lot of things for me. I'm not used to working by other people's rules, and there are a lot of rules when you work in an active mine and a construction site.
It's even an air shower. You have to walk through.
Yeah. So the air shower is awesome. It looks like a Superman telephone booth. And you walk in and you close the doors and you pull your shirt off and this S appears on your chest, but it's a whirlwind of, it is literally a whirlwind of air that blows the dust off of you. They are so concerned about dust.
And tacky mats you have to walk on in and out of doors.
Oh, we can't, I don't think I can get the camera over there. Every single door as you walk through it, there is a blue welcome mat, except it's actually made to be sticky so that the little bit of dust that's generated as you walk is stuck on those tacky mats.
Yeah. They peel off, so you have like 30 layers before you have to change it out.
Yeah. But dust is a really big deal down here. You shower to come in, you put on head nets, you put on a bunny suit. They've got the tacky mats everywhere.
And if you're with a real sensitive experiment and you have a beard, there are beard nets as well.
Yeah, and then the other thing is when you have this and the beard net, every time that you work, you have to ask somebody if they want fries with that.
Just you. Only you, Dr. Merritt.
Probably just me. I mean, the biggest thing for me was just not only actually having to obey the rules. I mean, I normally have a cup of coffee at my fly station, and there is no cup of coffee at the fly station here. But you know, we've got safety goggles, we've got a hard hat. When you come to work in a mine elevator. John and I are both certified deep underground mining contractors, and as a geneticist, as a genomicist, I never really anticipated that I would be an industrially certified contractor. But to work in this field site, you have to be prepared to be a miner. And when you go through the training, I mean, these guys are very serious about it. And they're like, if you're going to work in a mine for 30 years, you will be in a disaster. Here are the things you need to know to stay alive. And I got to tell you, as a professor, there are very few occasions where I have to think about, you know, what I'm going to do that day that I need to do to stay alive. And we get trained on this is how your safety belt works. This is how to fix a gas leak. This is how to use an air pipe to get air into a refuge if you're stuck underground.
And we have a bunch of army rations in case we are stuck here.
Boxes and cases.
Yeah.
It's a very different place than working on the surface.
It sounds like it.
Go ahead.
Oh, go ahead, Brenda.
I'm going to say the pieces of equipment that you see, like what it all takes to build an experiment is astronomical, because it's like a big silo, and inside you have a big vessel with these things sticking out and copper, and then big bladder, and it's filled with water, and it takes, you know, it's a lot that goes into putting together one of these experiments, and every piece of equipment is come through and ultrasonically cleaned and
ultra clean.
Yeah.
The amount of time and money that it takes to run SNOLAB. SNOLAB is about a $500 million investment. And that's a really big deal. And honestly, I mean, one of the things that I've talked about before is how seriously we have to take the investment that we're making in our research. There's a lot of money that goes into supporting this research, and we need to make sure that we're doing the best that we can with that. And whether that's, you know, pushing the envelope of particle physics to the point where you win a Nobel Prize, or understanding the biochemistry and physiology of working deep underground, those are real responsibilities. And I honestly feel like the work that John and I are doing down here has the possibility to make mining a healthier workplace, because we now are beginning to understand the biochemistry, the metabolism, the physiology, what it means to actually work underground.
Yeah.
And that,
you weigh more underground.
is that right?
Yeah. You know that gnome that they had all going all around the world?
Yeah?
Yeah,
They took
he came down here. Yeah. Yeah.
You're not talking about Stephen Hawking?
No, he was here twice.
He was here, yeah.
Yeah,
Okay.
he was here twice too.
Yeah.
Yeah.
It's an amazing place.
Yeah.
So we did, we also promised we would talk a little bit about the metabolomics today, and we've talked a lot about SNOLAB so far, and it's been wonderful. But I would be remiss if I didn't ask you a little bit about the experiments you're running, maybe show us what those flies look like, and also talk about any preliminary results that, that you have.
Absolutely. Let's do that. All right, why don't we thank Brenda?
Okay.
Thank you so much, Brenda. It was really, really interesting.
All right, that's great. Thank you
so
much. So what we are interested in is what happens to metabolism under different conditions. And I mentioned that we do things like starvation or desiccation. Working deep underground has certain physical challenges, and we think that one of those challenges is simply the high atmospheric pressure. So when you get 2 kilometers underground, the atmospheric pressure is about 20–25% higher than it is at the surface. And we think that that puts a physical stress on your body. And we know that when you come down to SNOLAB and go back up to the surface, you're going to be more tired than you are on a normal day. And so what we've been doing is trying to take a genomic and a biochemical approach to understand where that stress is coming from. So we take flies. Flies have a very similar biochemistry to humans. And so we've got a series of flies here. Come on, I can show the flies. There we go. I got them. Those are flies. So we work with flies because essentially the biochemistry of flies is identical to the biochemistry of humans. But we can bring down flies this rack of flies has 800 flies in it. We've got 40 vials of 20 flies. You can do the math. That's 800 individuals. And what we're trying to do is understand really broadly what happens to the metabolism and biochemistry of an organism. And if we were to study me, we were to study John, we were to study Brenda, Stacy, if you came down and joined us, we would all respond to that time underground, that higher pressure, in a slightly different way. And those differences are really interesting, but as we begin to understand the system, what's really most interesting are the consistencies. How do we all respond in the same way? Well, if we do that with 4 individuals, we have a very limited experimental power to allow us to do that. With 800 individuals, that gives us a lot of experimental power. Each of those individuals is responding slightly differently, and what we're looking for is how are those individuals responding in a similar fashion. And then we use metabolomics to try to understand that from a really broad biochemical perspective. So if you think of a gene and you think of genomics, so if we study an individual gene, we're doing genetics. If you study thousands of genes, all of the genes in an organism, that's genomics. Metabolomics is taking the same approach to metabolism. So instead of studying this metabolite or that metabolite, we're going to study thousands of metabolites at a time, and we're going to take a metabolomic approach. And so that's what we've been doing with the flies. And from our pilot experiments, we find that flies respond, about 10% of the metabolites in flies respond to being deep underground. And the interesting thing that we'll be working on now, honestly, probably for the next decade, is trying to understand what that 10% means. Which individual metabolites are changing, which metabolites are going up, which metabolites are going down. Some fraction of those are going to be healthy. They're actually they're helping you respond to being deep underground. And some of those are going to be a negative response. And so you can think of sort of an immune response that's making you healthier versus an allergic response that's a negative response to the environment. And one of the things that we're going to be working on, and that we are working on, is trying to distinguish between those 2 different kinds of change in the fly. And then the other thing that we've been able to incorporate in just this week is we're taking an exercise approach. So we take flies like this that are sitting on a countertop and they're relatively inactive, but we can also put them on a treadmill. And I think John is desperately typing start into the treadmill. And we'll see if I can turn the camera here. And there's John. And where are the treadmills? They're the treadmills. So here is a fly treadmill. And what that is gently rotating the vial. And when you rotate the vial, the flies tend to walk. And what John was able to show this summer is with as little as 2 minutes and really pronounced effect in about 15 minutes, you can actually get an exercise effect in that fly. And if we want to understand the biochemistry of working deep underground, we need the flies to work. And so all we're doing is physically making those flies more active and then looking to see how their metabolism changes in response to that activity. And how does that differ here from the flies that are 2 kilometers above us in SNOLAB?
I bet that our viewers were not expecting to learn how you can take your fly for a walk.
There you go.
It's really interesting. So you can make them exercise essentially and really mimic what it is that you're looking to learn about.
We were really sort of proud, okay, I was really proud of the fact that we came up with this idea of a fly treadmill and this idea that we could actually physically exercise the flies. And I was talking to a friend of mine about it, and she said, well, you know, I'm publishing a paper about that. And independently, she had come up with another fly treadmill. And as we were talking at a meeting, another lab came up, and they have another fly treadmill. So this is sort of a thing. And there are 3 of us that independently have different fly treadmills, different ways to exercise flies. And I mean, it gives you an idea of how many people are working on studying metabolism using that fly model.
Right. And so there's actually a reason why our grade 11 students are learning about Drosophila, and that it's actually still current in research.
The Drosophila research right now is amazing. There is some really fantastic work being done on heart disease and trying to understand the link between obesity and heart disease. So everybody, I think, is fairly comfortable with the idea that obesity leads to heart disease, but it ends up that metabolically, we are only beginning to scratch the surface of understanding what the tie is. And one of the best models for that is flies. And we can actually go in genetically and unlink those. And you can create obese flies, and those obese flies get heart disease, but you can genetically engineer those flies so they become obese, but then they never get heart disease. And that's beginning to tell us about the genetics and the biochemistry that connects those 2 pathologies. How and why do you go from obesity to heart disease? There's some really interesting work in autism. There's been some work in a number of different behavioral disorders that are all based in flies. It's a very simple model. It's an easy model to keep in the lab, but it's an easy model to do a lot of really fundamentally interesting research in.
Very well. Well, unfortunately, we have run out of time. We're already 4 minutes over, so I really need to wrap it up for our students who may need to go on to another class. Is there anything else that that you would like to say in terms of advice for students who may want to pursue research or anything along those lines?
I will make it really quick because I understand we're over time. SNOLAB is one of the most amazing places I have ever worked. It is just like physically it's amazing, but there are also things like there are Nobel laureates who happen to walk around because they do their research here. And we're here because of a really interesting idea that I had one afternoon reading somebody else's work. And it's a very new direction for us. It's not the way that our lab traditionally studies things. So as you develop as a scientist, one of the best pieces of advice that I ever had was look for the new and unusual things and take some risks, try some stuff that are different, you know, go off in different directions. And it could very well lead you to a really interesting genetics lab located 2 kilometers underground at the base of a mine shaft.
Well, speaking of new and unusual things, next week on PIR live event, in January, next week, enjoy your holidays, everyone. In January, we are going to show you RoboGut, a benchtop bioreactor that mimics the environment of the human colon to study the microbiome. So you can find more information on that on our website at pirweb.org. For now, thank you very much, Dr. Thomas Merritt, for joining us all the way from 2 kilometers underground at SNOLAB. Thank you to John as well, and to Brenda for giving us even more insight into what that's like and showing us a Drosophila treadmill. So thank you so much for sharing your expertise with us today.
Thanks for joining us. It was great to have everybody here.
All right. Have a good one, everyone. We'll see you next time. Bye-bye.